The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone. My name is John DeBella, Vice President of Marketing and Sales, and on behalf of Simulations Plus, I welcome you to today's webinar, which will focus on the use of global sensitivity analysis and physiologically based pharmacokinetic modeling to improve bioavailability during lead optimization. Dr. Eric Martin, Director of Computational Chemistry at Novartis, will be presenting today. An opportunity to ask questions will follow Dr. Martin's presentation. You may either send your question using the questions pane on your control panel, or you may ask your question directly using the hand-raising icon. If you're using a telephone to listen to the call, please be sure to enter the unique audio PIN displayed when you joined. When you enter your audio PIN, this enables us to unmute your line so that you can be heard. This webinar is being recorded for playback at our website, www.simulations-plus.com. Before Dr. Martin begins, Dr. Mike Bolger, Chief Scientist at Simulations Plus, will provide some background to this project and formally introduce Dr. Martin. It is now my pleasure to turn the presentation over to Mike.
Okay, well, I'm really pleased to be able to introduce Eric Martin today for today's webinar. Eric earned his PhD in physical chemistry from Yale in 1983, and since then he's worked in the field of computational chemistry as it relates to both herbicide and drug design. He is best known for starting the field of combinatorial library design in 1993, and his recent research focus is on protein family virtual screening in order to predict both biochemical and cellular activity with the accuracy that's comparable to experimental high-throughput screening. In 2011, he was awarded the lifetime title of Novartis Leading Scientist for this work. His latest research applies global sensitivity analysis of physiologically based pharmacokinetic calculations to identify the sensitive compound properties to improve bioavailability for individual medchem series during lead optimization. I've known Eric for many years as a valuable collaborator and early adopter of our software. Eric contacted me about 3.5 years ago to ask if I would be willing to serve as an external postdoc advisor for a project funded by Novartis Institutes for Biomedical Research to test the use and validation of PBPK simulations in early discovery and candidate selection. We also invited Ian Haworth from USC and our own Bob Clark to act as external advisors. It took some time to get the funding approved and to select a candidate, but I'm pleased to say we found a great person for this project. Pankaj Daga, who earned his PhD in medicinal chemistry from University of Mississippi, has done a great job learning how to use the software and incorporating his own background in modeling, building, and QSAR. And so with that, I'd like to turn it over to Eric for what I'm sure will be a great webinar. Thank you.
Thank you, Mike. I appreciate this opportunity to present the work that I and my many coworkers have been doing in improving bioavailability during lead optimization using global sensitivity analysis of physiologically based pharmacokinetics modeling, and primarily, of course, Pankaj, who is the person who's actually done the real work behind this. And the problem we're trying to address is the dilemma faced by a medicinal chemist when there's an exposure problem, in this case, too little oral bioavailability, because there's a tremendous number of input of compound properties that they might consider trying to modify in order to improve the bioavailability. But any multi-parameter optimization is a combinatorial problem, and the difficulty increases exponentially with the number of different properties that have to be addressed. And therefore, it would be really helpful to the medicinal chemists if we could point out 2 or maybe 3 properties that were the most important ones driving bioavailability for their particular medchem series, and in fact even for particular lead compounds that they might be interested in within their medchem series. And so the real question we're trying to help them answer is what compound to make next. And there are a number of approaches to use computers to help with predicting pharmacokinetics. Allometric scaling and traditional pharmacokinetic models are well-established methods, but they require animal experiments in advance, and so they can't be used on compounds that haven't yet been synthesized. Another approach is to use quantitative structure activity or structure property relationships, but because those are purely statistical empirical models, they require a tremendous amount of input data, and it's going to be rare, if ever, that a lead optimization project will have enough PK data to parameterize such a model, even though that model could then be used on virtual compounds. However, physiologically based pharmacokinetic models, which simulate the entire process of the compound dissolving in the stomach, passing through the intestines, possibly precipitating or dissolving further as the pH changes, being absorbed through the intestinal wall into the portal vein, and then undergoing first pass extraction in the liver, and then being distributed through all the tissues before it's eliminated by excretion or metabolism. That physiological underlying model doesn't require nearly as much data to tune it to work well for a series of compounds and to demonstrate that it's actually working. And so this is a good method to use at this lead optimization stage, and it has the further advantage that the model inputs typically are the very properties that the med chemists are already thinking about in order to try and improve the bioavailability. Physiologically based pharmacokinetics, of course, has been around for quite some time now, but it's typically applied to just a few individual advanced compounds. We use it at Novartis most commonly for predicting human doses or dosing schedules for clinical trials. It's also used to prioritize late-stage compounds in lead optimization that would be sent on to expensive animal studies. Usually in this case, the inputs to the simulations are in vitro experimental data, or in some cases global QSAR models. And in some cases, it's accompanied by local sensitivity analysis, and that's illustrated by the three pictures on the right. So the most local of sensitivity analyses would be if you did a simulation of the properties of the compound of interest and then maybe perturb each of the properties one at a time a little bit to see how stable the model is with respect to those perturbations. Or in some cases, I've seen published examples where there have been a number of larger perturbations, but still only done one property at a time. So those are local sensitivity analyses. For a lead optimization on a whole medchem series, a different set of requirements. All of the inputs have to be calculatable from structure because the compound hasn't been synthesized yet. There's an advantage in that all being from the same medicinal chemistry series, local QSAR models can be built and used specifically for this problem. And the sensitivity analysis now has to be global sensitivity analysis, and that's illustrated in this last box where this is the rectangle is supposed to be a compound property space, and the dots here represent simultaneous variations of all of the input properties to cover the full combination of possibilities and get a response surface of bioavailability versus all of those property combination. So our approach is illustrated here. The first step is to assemble the input properties for all of the compounds from that medchem series which have PK data. And initially that'll be done with as much experimental data as possible, but then it has to be confirmed that it can be done with data that's calculated just from chemical structure. Then run GastroPlus simulations on all of those sets, possibly adjusting the way the calculations are done in order to give a good, faithful prediction of the exposure and, in this case, the bioavailability. Once it's confirmed that the models are working well and are reliable, then the next step is to identify and then sample the compound property space of all of the active members of that medchem series. and then run thousands of GastroPlus simulations on those combinations of properties, and then do a statistical analysis, in our case using partial squares regression, and look at the coefficients, and the largest coefficients then will correspond to the small number of properties which have the most influence on the bioavailability, to which it's most sensitive. And then the final step, which I won't be talking about, we haven't really done much there yet, is the then design of compounds that simultaneously optimize activity as well as the couple of properties that were pointed out from this analysis in order to try to maintain activity while improving the exposure. So I'll start out by just talking about this first stage of validating the models for each Medchem series. And I'll have two examples. The first one is a published data set from Merck for dipeptidase 4 inhibitors. They published rat PK data for 49 DPP4 inhibitors. And so we ran the simulations using the clearance data that was available in that paper, which was plasma clearance from the rat experiment. And when we compared the observed bioavailability to the predicted bioavailability, the correspondence was not very good at all. The vast majority of compounds were underpredicted. They were predicted to have very little bioavailability. And we thought that might be due to the use of plasma clearance, and we have internally a QSAR model, global QSAR model for intrinsic clearance. And so we repeated the calculations now using the predicted intrinsic clearance because intrinsic clearance was not available in the papers. And in this case, found that most of the compounds, the bioavailability was overpredicted. So neither one of those methods worked very well. And looking into the literature, it was reported that members of this series undergo renal clearance, and that would explain the disconnect, because if the plasma clearance wasn't just liver metabolism but also included renal clearance, then we would have over-predicted the amount of first-pass metabolism in the models, and therefore that would explain why using that those experimental data underpredicted the bioavailability. So to get around this problem, Mike pointed out that there's an ability within GastroPlus to manipulate, to optimize one of the input parameters in order to best reproduce the output parameter, in this case the bioavailability. And so from the 49 compounds, we picked 37 at random and then applied this method and found the ideal, what we're calling fitted systemic clearance, that would perfectly reproduce the observed bioavailability in the GastroPlus simulations. Of course, we can't use that on new compounds. So we built a local QSAR on those fitted systemic clearance values. And then we used that local QSAR to predict the systemic clearance for all 49 compounds. So that included both the 37 compounds in the training set and also the 12 compounds, the 25%, which had been set aside to validate the quality of the QSAR model. And when we then ran the simulations, those are shown in this graph, the observed versus the predicted. 68% of the compounds, the bioavailability was predicted within a factor of 2, and 85% were predicted within a factor of 3. And if you can see closely, the red stars are the compounds that were not used in training the local clearance QSAR model, and they are predicted about as well as the ones that were actually used in the fitting, and so this gives us confidence that we should get similar results if we applied this to new compounds that the chemists might suggest. We then tried another example This was an internal data set of 61 kinase inhibitors from a single Medchem series, for which we had quite a bit of in vitro and PK data. We first ran the simulations using as many of the measured inputs as were available, plus the experimental plasma clearance. And when we looked at the plot of observed versus predicted bioavailability, again, we found that a number of compounds were underpredicted. But in this case, we did have measured in vitro intrinsic clearance. And when we used as many measured inputs as properties as possible, including the intrinsic clearance, then we found that right out of the box, we got pretty good results. 65% of the compounds were predicted within a factor of 2, and about 80% were predicted within a factor of 3. We then used all in silico inputs from global QSAR models, including the intrinsic clearance, and there we found that we actually got quite similar results, 68% within a factor of 2 and about 80% within a factor of 3. And finally, we used in silico inputs from global QSAR models, plus again did the fitting of the systemic clearance. And in that case, we got remarkably good results. Now 85% are within a factor of 2, and almost 95% are within a factor of 3. And so this method of using a fitted clearance seems to be a good general method if you don't have the available input data, and gives an improvement even if you do have pretty good results using available experimental input data. So now we've convinced ourselves that for at least 2 Medchem series, the GastroPlus simulations are giving us good enough results to evaluate to try and determine the sensitive parameters. So the next step then is to identify and sample the property space available to active members of this Medchem series, run the gastro simulations on enough combinations to sample that space and identify the most sensitive properties. There are some complications in running global sensitivity analysis on chemicals because global sensitivity analysis is a well-established method, but most typically it assumes that all combinations of input properties are going to be valid. For example, if you're optimizing a chemical process, the properties might be the temperature and the reaction time, maybe the molar excess of one of the reagents, the amount of catalyst. And you can just take any combination of the temperature of those 4 properties and run the reaction in that way. But that's not true for chemicals, because many combinations are just going to be chemically invalid. For example, shown in this plot here for log P and solubility, and this is actually for one of the Medchem series, you would like to sample the whole space shown by the orange dots, but if you look at the combinations that actually occur, you find that there aren't compounds that are both hydrophobic and highly soluble, not so surprising, and there aren't compounds that are both hydrophilic and very insoluble. And so the first job is to figure out how to come up with only the realistic combinations of properties that are chemically meaningful. And then in addition, some of those chemically meaningful combinations of properties are going to lie outside the range of properties that are expected to be available to active members of that Medchem series. And so that subset of the chemistry space is represented by this brown rectangle. And so what we need to do is identify, then carve out everything outside that brown rectangle, and what's left we should then use in the simulations. One thing this means, though, is that a lot of commonly used global sensitivity analysis methods, like one of the most popular Fourier amplitude sensitivity testing, aren't going to be available because they require a very specific sampling pattern. So how do we get this set of chemically meaningful, valid combinations of properties? The approach we took was to start with the ZINC database. This is a public domain database of 16.5 million drug-like compounds, that is, compounds that fit Lipinski-type rules. And then we used ADMET predictor to predict the GastroPlus inputs for all 16.5 million compounds. And that then gives us this realistic property space, or at least a sampling of this realistic property space, because it won't then include any chemically meaningless combinations. And then we look at the range of active properties, of properties for what we consider to be plausibly active members of the Medchem series, shown by the brown rectangle, and we filter through those, and we're left now with 3 million compounds that are fit within that range. That's still too many to run simulations on. Some of the input properties are correlated, and so we first perform principal components analysis on the property space, and then we sampled it with a grid to get it down to 4,000 compounds. And then we took those combinations of compound properties, and which we now considered physically valid, chemically valid, and accessible for this Medchem series, and ran the GastroPlus simulations on all of those. So that was one problem we had to overcome. Another problem is that a lot of drugs have many ionizable groups. And that is a complication, first of all, because if some of them have a lot of ionizable groups and some of them have just one or two, how do you, for the inputs to the sensitivity analysis, say which ionizable group corresponds in the complicated compound to the ionizable group in the simpler compound? And the other thing is that then, even if you could do that and say, okay, there's 5 ionizable groups here, you've got to adjust them in this way, that would make a very complicated message to give to the medicinal chemist. It wouldn't be very helpful. But we discovered that we could get almost the same results that we did by using all of the pKa's by using just 2 pKa's, the ones immediately above and below the native pH. And so here's an example of a drug that has quite a few, I think 5 pKa's. Here's the pH versus solubility curve using all of those pKa's. And here's the one you get that's, if you only use 2, the ones above and below the native pH. And you can see that it's not exactly, but it approximately reproduces the same rough shape of the curve. And when we ran GastroPlus simulations using only 2 pKa's for over 500 marketed drugs, we found that the correlation coefficient, R squared, was equal to 0.93, quite a good correlation. There were a few outliers, but for the most part, that was adequate. And so that made it simple to decide how to line up the ionizable group, the pKa values, as well as making a much more simplified message to relate coming out of the calculations. A third problem is that the units that are typically used for these properties are not always well distributed, and the units themselves, the values are arbitrary, how they're represented. And so that would bias the relative importance of the coefficients. And so for each of the properties, we had to look at the distributions. For example, here's the distribution of intrinsic clearance. And in order to get a fair distribution, we had to take the log of that value, transform it, and then for all of the values, we z-scaled them. That is, we subtracted the mean and divided by the standard deviation so that they would all be on the same units and could be fairly compared to each other. Finally, when the inputs are correlated, if you try to model them with, for instance, multiple regression, the coefficients are not stable. If you have 2 values which are highly correlated, one of them could end up with a very large positive coefficient, and the other could end up with a very large negative coefficient. And they would largely cancel each other, and the model would be fine. It would give good predictions, but interpreting the coefficients as the importance of those properties, you'd be misled, because it would look like they were really big when in fact they were mostly canceling each other and the overall contribution is very small. And partial least squares regression is a statistical technique that takes that correlation into account and forces the coefficients to move together a little bit in the same direction instead of moving a lot in opposite directions. And furthermore, as I mentioned, the processes of ADME are complex and nonlinear. If we partial least squares is generally a linear model, but you can give it nonlinearity by including higher order polynomials in the linear combination. And we found that if we did quadratic partial least squares, in both cases we got good correlations between, we got good fits of the predicted activity based on the inputs. In this case, the correlation coefficient was 0.88. So we felt that that was a good enough model that we could interpret the coefficients. The other thing to do is to prove that the coefficients don't change when you take different subsets of the data. So we did 1,000 steps of bootstrapping as a way of subsetting the data and rebuilding the models over and over again. And this plot here shows the stability of the coefficients. So coefficients are shown on the left, and it's the width of these marks. They're actually box plots. And for the most part, the coefficients are so stable that you can't even see the width of them. You can see a little bit for log D, the box plot-like shape to it, and for log D squared. But for the most part, these coefficients are rock solid, and so there isn't an overfitting going on that would make it the coefficients unstable and make it hard to understand what they mean. So that's the method, and now we can apply it to our two cases. And looking at the bioavailability for the kinase series in the left plot versus the DPP4 series in the right plot. First of all, for both of them, the most important property is intrinsic clearance. But after that, in the kinase series, what are shown here are the coefficients, the bars, lengths of the bars are the size of the coefficients or the sensitivities of the other properties which were at least 25% as big as the intrinsic clearance. And you can see that the distribution coefficient is almost as important as the intrinsic clearance, and the water solubility is also pretty important, as well as some influence of the blood plasma ratio. And there's also some curvature in the log D. It gets more important as it gets really big, and also an interaction between the intestinal permeability and the water solubility. So if you were talking to the medicinal chemist, that's what you would tell them to focus on, is that you have to make sure that the intrinsic clearance is not so fast that you lose the compounds in first pass extraction, but also that the lipophilicity is high enough that they can permeate, and that the solubility is high enough that the compounds are available to be absorbed. Whereas for the DPP4 series, you give them a very different message. The reason the few bars that I've shown here are grayed out on the right is that this blue line, which is pretty faint, you might not see that, is the 25%, and no other input property besides the intrinsic clearance reaches that standard. And so the main message you would give to the chemists in this case is that you've just got to improve the intrinsic clearance. Nothing else you do fiddling with the compound properties is going to help enough to give a significant improvement in the bioavailability. If instead of looking at the bioavailability, if you just look at the fraction absorbed, now the intrinsic clearance is taken out of the picture, and you still see some differences. Some new properties show up. The intestinal permeability shows up in the kinase series, and also a little bit of influence from the lower pKa. Whereas in the DPP4 series, solubility and intestinal permeability are important, but the log D doesn't show up at all, and the pKa only shows up as a modification of the solubility. So these are the different kinds of advice that you would give to medicinal chemists working on these different projects as the result of this analysis. Also, within a project, within a Medchem series, different compounds lie in different places on this compound property bioavailability response surface. And so if there's a particular compound in that series that they like and that they're trying to tweak in order to improve the bioavailability, but otherwise keep many of its properties, you would give them different advice for different compounds. For example, if we look on the kinase series at the log D versus the water solubility, for this particular compound, the advice would be that improving the solubility in this direction isn't going to help at all. It moves right along a contour line, whereas increasing the log D is the way that you could improve the bioavailability. Whereas for this other compound, the two log D and log solubility interact with each other, so you could actually improve the bioavailability by going at an angle across that surface. That is, improving both the solubility and increasing the lipophilicity at the same time. And that might be advantageous because you might be able to get away with a smaller increase in log D when an increase in log D might be undesirable for other reasons. Similar analysis looking at two different compounds and their log D versus the blood plasma ratio. Here's a compound where the distribution coefficient is the only thing that matters, that the blood to plasma ratio doesn't matter, whereas here's a compound where, again, they interact. And looking at solubility and the blood plasma ratio, you got a lot of different results for different compounds. For this compound, it's influenced by the blood plasma ratio and not the solubility. For this compound, the blood plasma ratio doesn't move along a contour, and increasing the solubility is what would make a difference. Here's an example where the shortest path is to try and improve both of them together. And here's an example where, although there are contours, they're close. That's just where it chose to draw them. In fact, it's just pretty flat, and neither one of them has a big influence. So in conclusion, we looked at two cases and found that for both of them, the GastroPlus models could be tuned to work for the entire Medchem series. And it could be used not only with in vitro inputs, but also with in silico predictions as the inputs, so that it could be used for designing new molecules. In difficult cases, or cases where some properties aren't available, we could use a optimized input parameter and then build a local QSAR on that and use that local QSAR to get the in silico prediction for that property. And by doing that, we found that the fitted systemic clearance worked extremely well. In some cases, a global QSAR for the intrinsic clearance also worked well in one of the cases. In neither case did the experimental plasma clearance work well for the whole Medchem series. Applying the global sensitivity analysis in order to find the sensitive compound properties for chemicals is a complicated matter. We were able to ensure valid property combinations by taking the combinations from a library of drug-like compounds. We found we could get around the problem of too many ionizable groups by picking just the pKa's above and below the native pH. Each property had to be examined in order to find the right transformation and scaling of the data to avoid bias so that the coefficients could be interpreted, and a quadratic partial least squares regression model gave a good fit and gave stable coefficients and got around problems of correlation among the input properties and the instability that causes among the coefficients. Finally, then, global sensitivity analysis did identify sensitive properties for each Medchem series, and they did differ for the two different series. Although intrinsic clearance dominated both of them, the octanol, the distribution coefficient, and water solubility were sensitive parameters for the kinase series, but not for the DPP4 series. And finally, the global sensitivity analysis identified specific optimization paths for the individual compounds within a series. The next steps, which we haven't yet accomplished, are to apply this to a project by actually designing compounds that then maintain activity while improving those properties. These were both retrospective studies where we had a lot of PK data available. We now want to try it in a live medicinal chemistry setting. And our approach we intend to use is to use some available evolutionary algorithms for perturbing molecular structures systematically and evolving them to improve, in this case, multiple parameters. And so commercial software, including Mo and StarDrop, have the ability to evolve molecules against some objective function, and that objective function can be a combination of a prediction of biological activity from, say, docking or a QSAR model, along with predictions of the inputs identified by the global sensitivity analysis. The initial evolution, because we want to do that through a lot of iterations, would probably be done on the PLS model itself, because that runs very quickly. But then the compounds that were identified, a large top fraction of the compounds that were identified, would then be put through the GastroPlus simulations to try and get the best possible estimate of how bioavailability they would be. So with that, I welcome your questions, your comments, your criticisms, or anything else you'd like to contribute.
Thank you so much, Dr. Martin, for a very excellent and insightful discussion. Once again, as a reminder, if you'd like to ask a question using your telephone, please use the hand-raising icon on the control panel and be sure to enter your unique audio PIN. There were a number of questions that came in through the questions pane, and what we'll do is try to hit those in the order in which they were received. The first question comes from Jaime, and it's related to the clearance which was fitted in one of the first examples. Was the CL fit calculated using the optimization tool in GastroPlus? And if so, what was the clearance adjusted to or against, bioavailability or some other endpoint?
Yes, it used the optimization tool in GastroPlus, optimizing against bioavailability, and that's why there was that perfect correlation then between observed and predicted bioavailability on the optimized properties. Although then there was scatter when we replaced those perfectly optimized properties with the ones that were the predictions from the local QSARs built on those properties.
Thank you so much. Another question comes in here from Jonathan. The QSAR that was developed for the Merck project, can you discuss a little bit about some of the descriptors that went into that QSAR model? Were they mostly physicochemical properties of the compounds?
No. So that was a global QSAR. It wasn't specific to the Merck. This is the intrinsic clearance QSAR, I assume. That was a global QSAR built on some 4,000 intrinsic clearance values from Novartis experiments, and it was a what's called an HQSAR, a holographic QSAR using the Sybil program, which is based on chemical substructures, substructural fingerprints, and a few properties, I think, as well. Although we've built models with an ADME predictor, we've built models with the R package on those same data sets, and found that the models are pretty comparable. We can get pretty comparable models from any of those approaches.
Thank you, Eric. Another question from Binfeng. In the kinase example, since most of the compounds are kinase inhibitors, what is the confidence to be able to use this model to predict compounds in other categories or in a global sense?
Well, if I understand the question correctly, to the extent that kinase inhibitors have a certain set of properties, they tend to be flat, they tend to have a certain size, they tend to have hydrogen bonding groups that can interact with the hinge. The range of properties that would be available to them would be a specific subset of properties. And we wouldn't think that the sensitivity analysis that was applied to that project would apply to, say, a protease project where the properties of the compounds are quite different. But the inputs to the model that we were using for the global sensitivity analysis, they didn't take into account the specific chemical structures. They were just things like the range of octanol water partition coefficients, of solubilities, the various range of pKa's, etc. And so to that extent, it's agnostic to the fact that they are kinase inhibitors specifically. I'm not sure if that answers the question. If the person who asked the question would like to follow up, then I'd be I could maybe say more.
I think you did a good job there, Eric. Thank you. Mike, I think this might be a question more aimed at you from Alex. Can you clarify the term native pH?
Yeah, the native pH that we can both calculate in ADME predictor and also in GastroPlus is representative of the pH of a solution when you take a molecule and dissolve it in water. So if, for instance, you had a highly soluble acid and dissolved it in water, it would drive the pH down. If you had a more weakly soluble acid, it would go into solution to a low extent. The pH would be driven down a little bit from 7, but not very much. And so we call the native pH that pH which is achieved at the saturation solubility of the compound, either acids or bases or Zwitter ions.
Thank you, Mike. Another question from Jaime. Can you comment on the software that you use for FAST, PCA, PLS, and bootstrapping?
So for FAST, we used R, but in fact we couldn't, and we did run that, but in fact we couldn't use the results because we couldn't find a way. That's where it occurred to us when we did that we were ending up with these physically unrealizable combinations of input properties. The partial least squares was also done in R. What were the others?
I apologize. I had disregarded the question. So it was FAST, PCA, PLS, and bootstrapping.
Yeah, all of those other statistical methods, all of the statistical methods were done in R. So the principal components analysis and the bootstrapping as well.
Thank you, Eric. Another question from Dennis. First, a comment. Very good talk. On slide 12, there's an excellent correlation. Among these 400-plus drugs, are some of them present in the training set of the pKa QSAR model?
That is a good question. Let me just bring that plot up. No, these are all marketed drugs. Oh, are they present in the training of the, I'm sorry, could you repeat the question to make sure I get the right answer?
Sure. So among these 400 drugs, are some of them present in the training set of the pKa QSAR model? This may actually be a question that Mike might want to address, if it's using the ADME predictor model.
Right. Yes, Mike would probably be best to answer this, but I suspect they would have been, yeah.
Yeah, I think that in the 400-plus marketed drugs, we, let's put it this way, our training sets for pKa contain many tens of thousands of ionization constants. I would say that a very small fraction are actually representative of marketed drugs, but wherever we have experimental data on marketed drugs, we do include them in the training set for the pKa models. I'm not positive about the specific 400 that we're talking about here, but you know, we try to cover as much chemical space as possible when we're building those pKa models.
Thank you, Mike. Another question coming from Marvin, was KPLS considered as an alternative to quadratic PLS?
That's a good question. That's right. If you use a kernel PLS method, then that will also take into account local models, more local pieces of the response surface instead of a global model. Because the goal here is to look at sensitivity coefficients, I think that a global model, I think that a model that's fitting globally on all the data with some amount of curvature is going to extrapolate better and interpolate better between those islands where we have data. That would be my personal bias, but no, we didn't try using kernel partial least squares.
Okay, thank you, Eric. A question from Warren. Why did you use ZINC as the compound property space?
Because it was available, and this is a postdoc project, and so we wanted to have as much of it be public and reproducible as possible. But other compound collections, like the internal Novartis collection, could have been added as well. The particular data sets were also added as well as ZINC from the two projects, the Merck project and the Kinase project. So ZINC plus those few other compounds to make sure at least the range of those compounds that had been already synthesized was covered.
Thank you, Eric. A question from Ting Ting. Out of curiosity, have you done a similar optimization analysis regarding oral exposure instead of bioavailability?
We did some initial work on oral exposure, and the results were not as good. The predictions for the entire Medchem series were not as good, presumably because there are some factors that cancel when you compare the oral exposure to the injected exposure. So as the initial points of trying out this methodology, we focused on where we had initial success, which was better with the oral bioavailability. We certainly intend to go back, especially for projects where that's the important property, and see how well we can do on that now that we have more experience.
Thank you, Eric. A question from Pallavi. In your opinion, how many molecules might be needed to do a reliable GSA?
That's a very good question. That's on Pankaj's on the short list of things that Pankaj is working on, where we're going to address that by taking these two projects that we've already studied and then dividing, randomly eliminating some of the available experimental data and see if we can get by with less and less and less. From what experience we have so far, we're thinking that something like 20 values would probably be needed, but we really want to run these calculations more systematically to find out.
Thank you, Eric. A comment and question from Ben. Nice job, Eric. What level of prediction accuracy might be needed for physical chemical property prediction tools to be useful in the GSA analysis?
So that's one of the reasons. That's another thing that I didn't talk about. That's actually another thing that can be done with this tool. Global sensitivity analysis and propagation of error analysis are very closely related. And so one thing that you can conclude from this is that if there are a few particular experimental inputs, or if you put compound inputs, property inputs that are sensitive, then those are the ones that are going to require the most accuracy. And if there are ones that are insensitive, then the accuracy doesn't have to be so high. And so it's really a function of the sensitivity of those properties for your particular Medchem series. And that points you to the fact that you might then go back to your analytical group and say, here it turns out that log D is extremely important, and we were using a global log D model, but maybe you can measure the log Ds now for our set of compounds to make sure that we're getting those as well as possible, and then we'll add those and make a better QSAR model to make sure we have the best data for those properties. But the sensitivity coefficients are, are essentially also an error analysis.
Thank you, Eric. Continuing with the theme of prediction accuracy, we have a question from Fang, and I think both Eric and Mike might want to comment. What is the prediction accuracy using QSAR predicted physical-chemical properties in the absorption phase of PBPK models? How about using experimental values?
in just the absorption phase without the clearance.
Mm-hmm.
We have looked at that. I don't think I have any backup slides or anything about that. Just trying to think here.
I think that when Pankaj did his ACS talk, I think that he might have mentioned something about the comparison with doing these predictions with in silico properties predicted for partition coefficient, for permeability, for solubility versus the measured properties from the Novartis database. And I think the results were pretty similar, if not better, with the in silico.
Yeah, so for the overall bioavailability, I have the plots here that show it. For just the intestinal absorption without the intrinsic clearance, yeah, my rough recollection is that the results were better for absorption than they were for bioavailability, because the intrinsic clearance is the hardest thing to predict. And the models are so sensitive to it, but I don't remember the numbers.
Thank you, Eric. Thank you, Mike. That is the last question that came in through the questions pane, and I do not see any hands raised. Oh, we do have another question coming in from Bin Fang, a follow-up question. Is it valuable or rational to build a model using the global compound database instead of a disease categorized database?
Oh, so the suggestion, I guess, is that build a QSAR model specifically for cancer compounds, or a QSAR model based specifically on, I don't know, respiratory compounds, because if those are inhaled drugs, they might fit a very different physical property space. My experience is that as long as you add the compounds from your specific problem to the global model usually does as well or better than a local model. And so dividing up the compounds into subsets based on particular disease areas for the compound properties, if that was the question, I don't think would give an advantage. Mike, you've done a lot of this kind of modeling too. What would you say?
Well, I would say that you guys are the first ones to really focus on these PBPK predictions for congeneric series. And I think that approach is probably going to be pretty valuable for drug discovery because you're not constrained to have to try to predict global properties. So I think, yeah, the global sensitivity in a series that is related to a particular program is probably the best way to go in applying these technologies.
Yeah, and for something like the fitted, a QSAR model for the fitted clearance, systemic clearance, that I think has to be a local model for your Medchem series. I think
right.
Additional compounds there would just add noise.
We are trying to, we're continuously trying to improve our ability to predict, intrinsic clearance based on global models and as large a chemical space as possible. But I think in the specific area that you're working in, some of these local models may be even better.
Thank you, Mike. Thank you, Eric. I again see no further questions in the questions pane nor a hand raised to ask a question via the telephone. So we will consider the webinar complete. I want to again thank Eric for the very clear, very insightful presentation today, and also Mike for his contributions. If you missed any part of today's presentation, the playback will be available again at our website, www.simulations-plus.com. Also, if you are attending the upcoming ISSX or AAPS meetings, please feel free to stop by our booth or visit with our scientists to discuss your projects and learn a little bit more about how our modeling and simulation technology can help. Thanks again for joining us, and have a wonderful day.